Good morning, students. We will continue the topic of lipid metabolism, competition number 4.2, and today's topic is digestion, absorption and transport of lipids along with abnormalities like lipid malabsorption. First we will see the dietary lipids, means the lipids present in our diet. The three main types of lipid present in our diet are trialkyl glycerol, phospholipids and cholesterol esters. Out of this, trialkyl glycerol is the major lipid present in the diet. So this trialkyl glycerol is hydrophobic, phospholipid is amphipathic. Amphipathic means it has both hydrophobic and hydrophilic parts, and cholesterol ester is again hydrophobic. So first we will study digestion. Definition of digestion is conversion of complex unabsorbable lipids into simple absorbable form is called as digestion of lipids. Digestion of lipid starts from mouth. In the mouth, the enzyme lipase is present. It acts on trialkyl glycerol and break the trialkyl glycerol into free glycerol and free fatty acids, three free fatty acids. This enzyme acts at pH 2.5 to 6. Therefore, in adults, this is not much effective. And second thing about this enzyme is that this enzyme break those trialkyl glycerol which has short chain fatty acid. Now, trialkyl glycerol with short chain fatty acids are mainly found in milk and milk product. Hence, the lipids which are present in our diet in the form of oil and fat, they contain trialkyl glycerol with long chain fatty acid. So this lipase is ineffective against this trialkyl glycerol, but it plays an important role in the digestion of trialkyl present in milk of babies. After mouth, we will see digestion in stomach. In the stomach, the enzyme gastric lipase is present. It also acts on trialkyl glycerol and convert it into free glycerol and three free fatty acids. It requires pH 5.4. This enzyme again ineffective in adults because in adult the pH of stomach is around 1.8, and again it will not effective against the triglycerol present in the oils and fats, because this gastric lipase acts on those trialkyl glycerol which contains short chain fatty acids. So it is effective in babies because the pH of stomach in babies is higher than adult, and milk contain trialkyl glycerol with short chain fatty acids. Now, digestion in small intestine. The major part of lipids are digested in small intestine. In the small intestine, two types of juice are present. One is bile juice and another is pancreatic juice. Bile juice is synthesized by liver, enter into the stomach through the biliary tree, whereas pancreatic juice is secreted by pancreatic gland, enters into the duodenum through the pancreatic duct. First we will see the role of bile. Bile provides alkaline pH, and this alkaline pH is essential for activity of pancreatic enzymes present in the pancreatic juice. Pancreatic enzymes require alkaline pH, but when food comes from stomach, it mixes with acid. It is called as acid chyme. So pancreatic enzymes are ineffective in acidic chyme, but that acidic chyme is neutralized by the bile and convert the bolus of food into alkaline, so that pancreatic enzymes can effectively act. The second role of bile is it contains bile salts. Bile salts are sodium and potassium salts of taurocholic acid and glycocholic acid. Now these bile salts plays an important role in the emulsification of fat. Emulsification of fat means in an aqueous medium the large fat molecule is broken down into small droplets, and this is essential because large fat drop cannot be digested easily by the pancreatic enzyme, but small fat droplets can easily digested with the help of pancreatic enzymes. So bile salts, by causing the emulsification of fats, helps in easy digestion of the fats. Now come to the pancreatic juice. Pancreatic juice contains fat digesting enzymes like pancreatic lipase, colipase, phospholipase A2 and cholesterol esterase. We will see the role of each enzyme. First, pancreatic lipase. Now this pancreatic lipase acts on trialkyl glycerol and break the trialkyl glycerol into free glycerol and 3 free fatty acids. It requires pH alkaline, which is provided by the bile. Now the enzyme colipase, as the name suggests, it is associated with pancreatic lipase and helps the pancreatic lipase to perform its action. Now this colipase is secreted as inactive zymogen by pancreas. It is activated by the trypsin. Trypsin is a proteolytic enzyme. Now after activation, this colipase binds to the trialkyl glycerol, and due to which the pancreatic lipase acts more effectively. So this is the action of pancreatic lipase. So pancreatic lipase first act on trialkyl glycerol, removes the one fatty acid from first position of trialkyl glycerol and convert it into 2,3-dialkyl glycerol. Later on, this pancreatic lipase acts on 2,3-dialkyl glycerol, remove one more fatty acid from the third position and convert it into 2-monoalkyl glycerol. In 2-monoalkyl glycerol, the fatty acid is attached to the second carbon of glycerol, and this is not hydrolyzed by the pancreatic lipase. That means the ester bond present at the second carbon of the glycerol is not digested by the pancreatic lipase. Therefore, another enzyme, isomerase, comes into play. This enzyme isomerase shifts the fatty acid from second position to first position of glycerol, so that 2-monoalkyl glycerol is now converted into 1-monoalkyl glycerol. Now this pancreatic lipase acts on 1-monoalkyl glycerol and removes the fatty acid from the first position of glycerol, and free glycerol is formed. Here you have to remember one point. The activity of pancreatic lipase is very fast as compared to activity of isomerase. Therefore, the formation of 2-monoalkyl glycerol is faster than its conversion into 1-monoalkyl glycerol. Hence, 2-monoalkyl glycerols cannot be converted into 1-monoalkyl glycerol in the given stipulated time of digestion. Therefore, at the end of digestion, near about 78% of 2-monoalkyl glycerol remains undigested. So these are the major end product of digestion of trialkyl glycerol. These are 2-monoalkyl glycerol 78%, 1-monoalkyl glycerol 6%, glycerol and fatty acid 14%. Thus, trialkyl glycerol are not completely digested in the GIT. Now the next enzyme is phospholipase A2. Now this phospholipase A2 acts on phospholipids, removes the second fatty acid from the phospholipid and convert it into lysophospholipid. Third enzyme, cholesterol esterase, acts on cholesterol esters present in the diet, removes the fatty acid from the cholesterol ester and convert the cholesterol into free cholesterol. Now this free cholesterol has OH group at third position, due to which this cholesterol gain an hydrophilic group in the form of OH. So cholesterol is now amphipathic in nature. That means it has hydrophilic part in the form of OH and the cyclohydroxyphenanthrin ring in the form of hydrophobic structure. Now the product of lipid digestions are short chain fatty acids, medium chain fatty acids, long chain fatty acids and glycerol. These are obtained by hydrolysis of trialkyl glycerol. Then undigested monoalkyl glycerol and 2 monoalkyl glycerol and 1 monoalkyl glycerol. Then lysophospholipids, phospholipids. These phospholipids are undigested and free cholesterol. So this completes the digestion of lipids. Now come to absorption of lipids. Absorption means transfer of digested lipid products from GIT lumen to the blood is called as absorption. It takes place in two phases. In the first phase, the digested product from GIT lumen are transferred to the epithelial cell, inside the epithelial cell. And in the second phase, this digested product from epithelial cell enters into the blood. So this is the diagram showing absorption of lipids. If you see, short chain, medium chain fatty acids along with glycerol, they directly absorbed by the epithelial cell, and from the epithelial cell they get directly transported to the blood. But the remaining products like 2 monoalkyl glycerol, 1 monoalkyl glycerol, long chain fatty acids, lysophospholipids, undigested phospholipids and cholesterol, they cannot easily enter into the epithelial cell. These are amphipathic lipids, but their hydrophilic part is very small as compared to their hydrophobic part. Therefore, they will not enter into the epithelial cell. Hence, these lipids get arranged themselves in the form of micelles. What are micelles? Micelles are nothing but the amphipathic lipids in aqueous medium arrange themselves in such a way that their hydrophilic part is facing outside and hydrophobic part is present at the core or inside the micelle. So when these micelles are formed, they are formed by combination of amphipathic lipids. They does not contain any strong bond. They have only hydrophobic bonds. Bile salts plays an important role in the formation of micelles by lowering the surface tension. Now once these micelles are formed, as I have already told you, their outer covering is hydrophilic. Hence, they enter into the epithelial cells easily. When these micelles enter into the epithelial cell, this micelle is broken down and all these lipids which are present inside the micelle or form the micelles, they get separated. So in this way, the lipids from GIT lumen enter into the epithelial cell. Now in the second phase, these lipids have to enter into the blood. Here there are some modifications occurs in these lipids in the epithelial cell. For example, long chain fatty acid, 2 monoalkyl glycerol, 1 monoalkyl glycerol get converted into trialkyl glycerol. In the epithelial cell, the glycerol 3 phosphate is synthesized from glucose, and this glycerol 3 phosphate then combines with 3 molecules of long chain fatty acid to regenerate trialkyl glycerol. Now this 2 monoalkyl glycerol and 1 monoalkyl glycerol also combines with 2 long chain fatty acid to regenerate the trialkyl glycerol. Similarly, lysophospholipid combines with fatty acid to form phospholipid, and cholesterol combines with long chain fatty acid to form cholesterol esters. So in this way, all the lipids which are present in the diet, that is trialkyl glycerol, phospholipid and cholesterol esters, get regenerated in the epithelial cell. Out of these 3, trialkyl glycerol and cholesterol ester are purely hydrophobic, whereas phospholipid is amphipathic in nature. Therefore, these lipids are not easily absorbed into the blood compartment. Hence, these lipids form a structure called as chylomicron. What is chylomicron? It is a lipoprotein, means combination of lipids and protein. At the core of this chylomicron, if you see this structure, at the center of the chylomicron you will see hydrophobic lipids like trialkyl glycerol and cholesterol ester. Just periphery to the core are amphipathic lipids like phospholipids are present, and whole lipids are then covered by proteins, and the main protein is ApoB48 protein along with ApoC protein. Why protein covering is important? Because proteins are hydrophilic. So when complete chylomicron is covered by protein, now that chylomicron is hydrophilic molecule, though it is a large, but it is a hydrophilic molecule because the outer covering is of protein. Now when these chylomicrons are formed by combination of lipids and proteins, they form some larger structure. Now this chylomicron therefore does not enter into the endothelial cells of gastrointestinal tract because they have small spaces. Hence these chylomicrons enters into the lymphatics of the GIT. In the lymphatic vessels there are large spaces in between the endothelial cells. Therefore chylomicrons easily enter into the lymphatics. From the lymphatic vessels, these chylomicron enter into the lymphatic duct. From lymphatic duct they goes to thoracic duct, and this thoracic duct then opens into the systemic circulation. So in this way the lipids are transported from GIT to the blood. Coming to the abnormalities in digestion and absorption of lipids. First is steatorrhea. Normally small amount of fat is excreted into the stool, but when large amount of fat, more than 6 gram in 24 hours, get excreted into the stool, that condition is called as steatorrhea. The causes of steatorrhea are, the most common cause is pancreatic disease. Whenever there is a disease of pancreas, the synthesis of pancreatic juice and its enzyme decreases. It leads to difficulty in the digestion of lipids. If the lipids are not digested, they are not absorbed properly and get excreted into the stool. Now the second most common cause of steatorrhea is biliary tract disease, means obstruction to the flow of bile. It may be due to various reasons. It may be due to gallstone, it may be due to stricture, cancer, tumor, etc. In this case, the bile which is synthesized by the liver does not enter into the small intestine, and I have told you that bile plays an important role in the digestion and absorption of lipids. Due to deficiency of bile, the digestion of lipid get hampered and they cannot properly absorb and excreted into the stool, leading to steatorrhea. Next cause is intestinal diseases. Here the absorption of lipid is hampered due to various diseases of small intestine, and hence that lipid is excreted into the stool. Two rare diseases are also seen. They are chyluria and chylothorax. Chyluria means excretion of lipid into the urine. How it happens? In some persons, it is found that there are abnormal connection between lymphatic vessels of GIT and urinary tract. Due to this abnormal channel, when chylomicrons enters into the lymphatic vessels, small amount of chylomicron enters into the urinary tract and excreted into the urine in the form of chyluria. Here the color of urine is white and patient generally complains white color urine after meal. The next condition is chylothorax. Again, abnormal connection is present between lymphatic vessels of GIT and thoracic region. Hence, small amount of lipids or chylomicrons enters into the thorax through this abnormal connection, which presses on the lungs, and the patient may complain cough after meal. These are rare diseases, but steatorrhea is very common disease. Now this is about digestion, absorption of lipids and its abnormality. Thank you very much.